Okay, so it's just 1 minute past 4 here in the UK. I see most people have signed on now. Welcome to our first scientific webinar series of the year. Had a little extended Christmas break, but we're back with more applications. And this is a publication that was done at the back end of last year, focusing on IL-6 and a T-cell bispecific engager. Just some housekeeping before we start. Obviously, if you could stay on mute while the presentation's going ahead, and we'll take questions at the end, so you can either write them in the chat as we go, or raise your hand at the end and we'll take those questions. Just bear in mind that the session today is being recorded, and thanks to Nassim and Neil to agree to do that. If you could just move on to the next slide, please, Nassim. So our two speakers today, Nassim and Neil, they previously worked together at Roche, but you can see here Nassim is now head of pharmacometrics at Lufthansa Biotech, and he previously worked at Roche, and before that I think you were at Sanofi. Yeah, that's right. Probably someone who is very well known to the audience is Neil Parrott. He's a distinguished scientist, predictive modeling and data analytics at Roche. Without further ado, I'll hand over to you guys, and you're going to talk about this publication, looking at the review of IL-6, impact on CYPs, and kind of the work that was done to get information into the label for glofitamab. So I'll pass over to you guys. Thank you again.
Thank you, Oliver. Thank you for having us here. I'm really happy that I'm here with Neil to present our work that was recently published, as you mentioned, on the impact of the transient increase of IL-6 levels following the administration of glofitamab. So before going directly into this specific example, we will give you a brief background about PBPK modeling of IL-6 in general. It was published before in other indications like autoimmune disease and ophthalmology, and then in cancer immunotherapies. We will end up with a specific example of glofitamab, and we'll share with you as well some reactions and interactions we had on this specific topic with health authorities. So before jumping on the topic, I wanted to give you a brief background, not only about glofitamab, but also about the contribution of clinical pharmacometrics and modeling and simulation to support the BLA submission of glofitamab. So glofitamab is a T-cell bispecific with a unique 2-to-1 format, meaning that we have got two binding regions of CD20 on the malignant B cells and one CD3 on the surface of T cells. So glofitamab is characterized with great efficacy because it's working very well in the most aggressive forms of non-Hodgkin lymphoma. Here it's in relapse refractory DLBCL, but this amazing efficacy comes with a cost, which is cytokine release syndrome, so called as well cytokine storm, meaning that there is an elevation of the levels of certain number of pro-inflammatory cytokines, and this comes with some symptoms that are life-threatening. So we need to consider that. And back in 2019, we used PBPK and exposure response models, exposure safety and exposure efficacy to come with and suggest the strong benefit of step-up dosing. that allow to maximize the efficacy while mitigating the efficacy, the safety, the main safety issue, which is cytokine release syndrome. And step-up dosing means that you could start the administration with a very low dose, that is a dose that is low enough so that you could stimulate the immune system, but keep the safety manageable. And then the following administration, you can go up and up. In this case, it was 2.5 milligram, and then 10 milligram, and then the full dose, which was 30 milligram. And in these higher doses, you could mitigate the safety and get your immune system tolerating more this cytokine increase. In parallel to that, we worked on PBPK modeling to assess the potential impact of the IL-6 on the CYP isoforms. So I will start by just briefly introducing the relationship between IL-6 levels and cytokine release syndrome. It started back here in 2014. You can see it with CAR-Ts, where in the left figure, you can see the severe CRS that are statistically correlated with IL-6 levels. And when for the patients experiencing severe CRS, you can see that the median value of the IL-6 levels are around 10 picogram per ml, while for patients not experiencing this severe CRS, they are more around 100 picogram per ml. So you can see this kind of threshold value of 1,000 picogram per ml that already appeared at the time. And here we are talking about severe CRS, meaning that patients need to go to the ICU. So it's really something that we need to consider and to find some mitigation strategies for that. Another example is blinatumomab. So blinatumomab is a bispecific, so it's CD19, CD3 bispecific. And what we want to show here is that CRS is more a first administration event because of what we mentioned. The following administration, you have got some tolerance of the immune system. And you can see it here. The blue curve is the cycle 1, orange curve cycle 2, and green curve is cycle 3. So first administration, you have these CRS levels with IL-6 levels that are around the 1,000 picogram per milliliter. Another example here is glofitamab. Again, you can see here that the grades of CRS from 0 to 4 with a trend of higher IL-6 levels when you go high in terms of CRS grade. But still, there is a big overlap between 10 and 1,000 picogram ml, meaning that there is IL-6 can be used as a surrogate of CRS, but still there is a between subject variability that is really high and make it difficult to really be predictive still. So there are some complexities here. Another way of presenting the same data here is that you can see there is a logistic regression model relating the maximum IL-6 levels to the probability of experiencing grade 2 and higher CRS. At the same time, when you connect the IL-6 Cmax levels with the CD20 receptor occupancy, you can see there is a trend. So we use these two figures together to come with this logistic regression model, connecting or using in the x-axis the CD20 average receptor occupancy to predict the grades of CRS. So with these different figures, there is a correlation that supports the dose dependency. Now I will give the floor to Neil to share with you some background about PBPK modeling of IL-6 levels.
Yeah, thanks, Nassim. So after some background on the IL-6 elevations with glofitamab, we'd now like to turn to the PBPK modeling, which was done to assess the potential for CYP suppression by these IL-6 levels. If you could go to the next slide, then please, Nassim. Thanks. So yeah, we'd like to start with this 2011 paper from Leslie Dickman, which described in vitro studies done to assess the potential increases in IL-6. So in this study, hepatocytes were dosed with different levels of IL-6, and then EC50 values could be estimated for various CYPs based on mRNA readouts. And these studies showed that the different CYPs were suppressed to quite a different degree. So with 1A1, we could see the least degree of suppression, whereas CYP3A4 was the most potently suppressed enzyme by the IL-6. And going to the next slide, please. Yeah, so a few years later, this paper from Macavaram and colleagues, CIMCIP colleagues, came out, and this was work done in a collaboration with Genentech at that time. And this was really the first study which explored the translation of these in vitro IL-6 suppression data to in vivo effects. And so this work in CIMCIP focused on building up an IL-6 compound file, which was then used to simulate these IL-6 elevations, either as a steady state level or as a transient increase. And in addition, the colleagues here collected literature data on the patients who were subject to elevated IL-6 levels and were also dosed with CYP3A substrates. And so using these clinical data together with the EC50 values which Dickman had reported, they were able to make simulations of the suppression and to compare their simulation of the suppression of CYPs and the reduction in CYP exposure with actual clinical data. And so this was like a verification of the IVIVE, the in vitro to in vivo extrapolation here. And this really opened up the way to using this type of modeling for predictions. And then based on this, a few years later, we at Roche, together with our colleagues in Chugai, collaborated with Krishna and the CIMCIP colleagues to apply this approach for one of our compounds, satralizumab. So satralizumab is an anti-IL-6 treatment for neuromyelitis optica, or NMO. And so what we did in this work was to collect clinical data on the pathophysiological changes in NMO patients, which in addition to the IL-6 elevations were involved reduced albumin levels. And so we put all of this physiological knowledge into the model, and then using the IL-6 file to simulate the potential for drug-drug interactions with CYPs, which could occur when the satralizumab was removing the IL-6, and thus you could see potentially an increase in the exposures. And we also looked at the potential for differences in different ethnicities using the standard CIMCIP population files in this work. Going to the next slide, please, Nassim. Yeah, so this is work done by Zhang and colleagues. And this is interesting, giving further support for the ability of this PBPK approach to predict the DDIs. because in this study they performed a clinical study with a cocktail of CYP substrates, and that study was performed in rheumatoid arthritis patients who have elevated IL-6 levels. And so the patients received a cocktail with midazolam, warfarin, omeprazole, and caffeine, and they received the cocktail first prior to and then after dosing with an anti-IL-6 therapy. And so what you can see from the plot here on the right-hand side is that after the treatment with the anti-IL-6 antibody, the CYP exposures, the exposures of these substrates were slightly reduced. And as you can see, the SimCYP modeling was quite nicely able to capture these observed changes with this cocktail of CYP substrates. Next slide, please. So this is another interesting study from Zhu and colleagues who applied the SimCYP approach for their T-cell engager, blinatumomab, which was already mentioned by Nassim. So blinatumomab causes a transient elevation of cytokines, including IL-6, IL-10, and interferon gamma. And as part of their study, they also performed an in vitro hepatocyte study with a cocktail of these elevated cytokines. And from that in vitro work, they could deduce that the suppression effect was mainly driven by the IL-6. And that then supported their PBPK modeling using the same type of approach and the IL-6 compound file, as has already been described in the other studies. In this work, interestingly, they also simulated out the effect of the time of dosing of the CYP substrate with respect to the blinatumomab dose, and found that peak suppression was occurring between 2 or 3 days after the dose of the T-cell engager here. So the suppression, as you can see from the plot, the suppression is you know, quite long lasting after the initial acute IL-6 elevations. Next slide, please. Yeah, so this is a more recent commentary paper, which is needs to be highlighted because it provides a very thorough review of various in vitro, in vivo, and modeling studies which have been performed to assess the impact of cytokines on CYP enzymes. And what you can see in this figure shown here, taken from the paper, is comparison of the actual clinical changes in Cmax and AUC for the various CYP substrates. Those are shown in blue here, and then also shown is the results from the simulation studies which have been published. So we can see here the McAvaron study, as well as the Jiang et al. Study and the Zhu study are all included in this figure. And in addition to the fact that you can see the simulations are capturing these changes quite nicely, it's also worth pointing out that the extent of these changes are all falling into the yellow and white regions of this plot, which correspond to a weak or no interaction, so less than twofold interactions in all these cases. Next slide, please. Yeah, and this is the last example I'd like to highlight, and this is from a very recent paper in CPT-PSP. This is work from Yuan Chen and colleagues at Genentech, and they developed the IL-6 model to capture the transient elevations seen after dosing of mosunetuzumab. So this mosunetuzumab is also dosed, as was mentioned, for glofitamab, is also following a sort of step-up dosing where doses are given of 1, 2, and then 60 milligrams on days 1, 8, and 15. And you can see that they were able to capture quite nicely those changes in the IL-6 levels, and then to simulate out the suppression profile for CYP3A4 here. And in addition to providing an estimate of the magnitude in the change of co-administered 3A4 substrates, the colleagues also found this simulation useful to guide the clinical monitoring of co-medications that could be recommended when narrow therapeutic index drugs are given in combination with this bispecific. So I think that is the end of my section for the moment. Nassim, back to you.
Thank you, Neil. Now I will deep dive into PBPK modeling of glofitamab-induced IL-6 transient increase. So it's basically what is described into the publication. I will give a bit more kind of technical and some insight about about the strategy that was followed here. Basically, the first question that was addressed here is how the transient increase of IL-6 levels would translate and would project into potential DDI risk with small molecules that could be administered with glofitamab. So from there, this is the methodology that was taken. First step is to incorporate these the in vitro CYP separation data into an IL-6 compound file. So it's basically the data coming from the Dickman paper, adapted from the publications from 2013 and 19 that were published on different indications. And then the second step was adaptation or model refinement and a couple of sensitivity analyses to adapt this model into what we have for glofitamab. So basically, it's about the population itself, using virtual Caucasian population with demographic characteristics that were matching the database we had for glofitamab step-up dosing patients. The couple of sensitivity analyses were to refine the PK parameters and the associated between subject variability to capture the IL-6 levels, the IL-6 concentration time curves. The second sensitivity analysis was more in terms of the input of the IL-6. So we needed to adapt the dose and the infusion duration so that we are able to really capture the concentration time curves. Because as opposed to autoimmune disease, for instance, where we have got higher IL-6 levels but steady levels, here we have got some peaks and troughs that we needed to capture. The third step was, of course, to have a look and to assess what kind of impact this transient elevation would have on the activity of the different CYPs. Of course, as it's a suppression, meaning that what Neil mentioned before in terms of the recovery of the CYP activity, it's something that we needed to consider and to pay attention to. The fourth step is then what we wanted to go for is the projections with different probe substrate of the different CYPs. We looked at CYP3A4 with simvastatin and midazolam, at caffeine and theophylline for CYP1A2, and S-warfarin for CYP2C9. So if I go deeper from a technical point of view here, as I mentioned, in terms of population, for the model refinement, we went from the Caucasian virtual population, and we really used the variance covariance matrix of the covariates, meaning the demographic characteristics from the step-up dosing patients of the glofitamab database. Then the first sensitivity analysis was to adapt the volume of steady state. We used this value that was similar to the one published earlier. We adapted the value of the clearance as well, so that it's similar to the one published by colleagues for blinatumomab PBPK model, where it was more adapted as it was a bispecific compound in cancer population. We increased as well the between subject variability, or the CV value, from 30 to 100% to really capture this high between subject variability we are observing in the IL-6 levels in the glofitamab patients. The second part of sensitivity analysis was, as I mentioned before, more about the input, about the dose and the duration. And as we realized that the IL-6 peaks were observed approximately 12 hours following the administration of glofitamab, then decided to go with a 12 hours IV infusion duration. And then we decided to go for 3 different scenarios. The scenario with a low level of IL-6, medium level, and high level. The reason for that is to make sure that we are capturing the best case and worst case scenario in terms of the DDI impact or risk that we are predicting. And one last point is about the priming effect. You know that this step-up dosing goes from 2.5 to 10 milligram to 30 milligram, but this does not necessarily translate in a proportional increase of the IL-6 levels. Because of the priming effect, we have got similar or even lower IL-6 levels at the second or third dose. And we decided then to go with the same dose independently, of course, of the scenario we choose. For example, if we go with the low level dose, we administer this low dose at first administration, the same dose for the second administration, and the same dose for the third administration. And that was justified by this priming effect that we do not need to go for higher doses. So this table is a table of the input parameters that you can find in the publication as well if you want to build from scratch the model into the SimCip simulator. So yeah, these figures, so if you focus on the blue curves, the blue curves in the 3 different figures are exactly the same. These blue curves represent the observations in the 2.5, 10, 30 milligram glofitamab step-up dose in patients. We on purpose decided to go with patients who did not receive tocilizumab because tocilizumab reverses the CYP suppression effect of IL-6. So to make sure that we do not induce any bias, we decided to go only with patients who did not receive any tocilizumab. And you can see here that the 3 different scenarios are really capturing the different levels of this IL-6 increase. So the low level and medium level capture most of the patients, in fact. And we decided to go for this high level scenario to capture the IL-6 CT profiles for the patients who experienced these levels close to this 1,000 picogram per ml of IL-6. So this is how the projections translate in terms of CYP3 of CYP activities. I've got CYP3A4 here, CYP1A2, and CYP2C9, and you can see that these dashed lines. So the green dashed line is following the scenario of low IL-6 elevation, and the orange is medium IL-6 level simulation, and the red one is the high level simulation. And you can see, for example, if I take the CYP3A4, that the very worst case scenarios, the high level simulation, at the end of the third administration of glofitamab, you can see that the activity of CYP3A4 goes down up to 50% of the baseline value, meaning that you could make already some predict here that for a prop substrate of CYP3A4, it will kind of double the exposure if we are at this, if the small molecule, if the prop substrate is administered at this level. It's less present here in the for the other CYPs, but yeah, it's you can see here how the shape looks like. So this impact of decreasing the CYP activity translate that way into, when we look at the different props, so simvastatin and midazolam for CYP3A4, I've got caffeine and theophylline for 182 and simvastatin for 2C9, and I'm only showing here the impact following the first administration. The impact is a bit higher when you look at steady state, for instance. Finally, here I'm summarizing in this table the AUC ratio following the first prop administration or over the whole simulation period. And when I say whole simulation period, it includes all these periods from the first administration up to the end of the third administration, in fact. And you can see here that the most important increase is associated with CYP3A4 substrate when we look at the high, so the worst case scenario, so high IL-6 levels. You can see that simvastatin and midazolam, after the first administration, the AUC ratio is predicted to be 1.75, more or less. And it goes up to almost 2 when over the whole simulation period. Now, if you look at the other CYPs, there is nothing of concern. The maximum value that we can see is 1.5 here. And if we focus on low to medium scenarios, where most of the patients sit, 95 of the patients are within this low to medium scenario, then there is no concern at all. The maximum value is 1.36, more or less, which is within the between subject variability of the PK of most of the small molecules we are concerned about, in fact. So as a summary from my side here is that the magnitude of the suppressive effect of transient IL-6 increase on the hepatic CYPs is less than 50%, and this will depend on the level and the duration of the cytokine elevation. Both parameters here are important. And the exposure of CYP3A4, CYP1A2, and CYP2T6 substrate increased by less than twofold in the worst case scenario. We have seen that the twofold relates more to CYP3A4 substrate for the very worst case scenario. And with that, no restriction on concomitant treatment with any other drugs, and only the consideration that may be given for potential DDI during the first cycle, as it's a first administration event in patients receiving concomitant CYP substrate with narrow therapeutic index, like monitoring toxicity for warfarin or therapeutic drug monitoring for cyclosporine. But anyway, it's in the label of those drugs to take care of that. With that, we'll switch over to Neil for us to give us some feedback about reactions and interactions with health authorities.
Yeah, thank you. So yeah, we'd like to conclude this by sharing some experiences with the interaction with health authorities on this work. So if you could go to the next slide, please, Nassim. Yeah, so according to the FDA guidance document, which is dated 2023, on drug-drug interaction assessment for therapeutic proteins, then for pro-inflammatory cytokine modulators, then we have two options here to follow. One is if we can provide scientific justification for no interaction potential, then that's fine. We don't need then any labeling language. However, the alternative then is conduct DDI evaluation, and if you look in the document, what constitutes an evaluation is it does not have to be a clinical study. It can also involve in vitro and animal work, as well as PBPK and PBPK modeling. And so I think that's essentially what we have described, or Nassim has just described, was the approach for glofitamab. And so following that, then the label that we obtained for this regarding the drug interactions was that for CYP substrates where small concentration changes could lead to adverse reactions, then the monitoring of concentrations should be undertaken when those are co-administered with glofitamab. And so the next slide is just to share what we got from the EMA, and this was more or less exactly the same recommendation in a little more differently worded, but the recommendation was that substrates with a narrow therapeutic index should be monitored. So this was the label that we obtained, and this was of course obtained without us performing any dedicated clinical studies with CYP substrates, but based on the modeling analyses that Nassim has shared. Next slide, please. Yeah, so I think it's clear that this work can inform labels without the need for clinical studies, but there's still this need for this cautionary language regarding the narrow therapeutic co-meds. The next slide, please. So in our interactions with health authorities during this work, we appreciated that there was still some lack of confidence in the predictions. This was based on limited experience existing to evaluate these projections, and was highlighted that there's still quite some uncertainty in the IVIVE for IL-6 suppression, particularly regarding the range of IL-6 levels explored. And I think here one of the limitations is basically that the clinical data against which the simulations can be verified is still quite limited. Authorities were also raising the uncertainties around the time course of the suppression effect, and so I think that's something that I think most of the studies we haven't really explored that that time course and the recovery after the suppression. So if we could do more there, I think that would increase the confidence. I think more shared examples of this approach, which are confirmed with clinical data, will certainly help. And there's been some more papers coming out in recent times. And another consideration is that this work has really very much focused on IL-6, and so there's still quite some gaps around the other cytokines, and if we can get more studies and data around those, that can also be helpful to increase the overall confidence. And I think with that, it should be our last slide. So we'd like to thank you for your attention and open up for discussion.
Thanks, Nassim and Neil. That was really interesting. We've had a question in the chat. If anyone else wants to either write down a question, let me know, or raise your hand and we'll get you unmuted and you can ask yourself. Ming Zheng has asked, and I think you addressed this a bit in your last slide, but could you comment on your experience on how well the in vitro CYP suppression translates to in vivo? If not as well, how could you suggest to improve?
You want to take that one, Nassim?
Yes, I think that, yeah, I came back to this slide, in fact, because I think it summarized well. Yeah, it's one of the points, one of the limitations we have in terms of how the in vitro CYP suppression that was well described in the paper of Dickman would translate in vivo. I think it's more related to how you would validate or qualify any of the models you would build. So coming from in vitro to in vivo, of course, the uncertainty without any validation is much higher as compared to when you start to collect some clinical data, and this uncertainty will shrink, or the certainty around what you would predict will increase with clinical data. And the reason for that, I think, is that we have got between subject variability that is high for some parameters. We can, I'm thinking, for example, about the turnover of CYP3A4 KDEG value. We know that we have some values in the literature. We have some similarities when you take the different PBPK platforms, and this KDEG value comes with some variability that might not be well reflected by your in vitro system, for example. And then when you translate that to the clinical setting, and of course, depending on the indication, as I mentioned, when I went to the non-Hodgkin lymphoma patient, I needed to increase the between subject variability of volume of distribution and clearance. This is one example, but you can imagine then that coming from one population to another, it's associated with different values or between subject variability of these parameters. And I think this is where the complexity comes in this kind of translation.
Thanks. I think you mentioned in the talk about how you looked at different populations and the effects, kind of linking into what you were just saying. Did kind of the regulators take any interest in that, or was that just something you did internally to look at across different populations, the effect?
Yeah, so for satralizumab, we explored Japanese and Chinese population files. The effects in the different populations were very, very similar. But we didn't get too much feedback from the regulators on that. For satralizumab, we didn't get the same labeling. We managed to avoid that monitoring statement for satralizumab. I don't know if it's related to the fact that that was an anti-IL-6 treatment. distinct from glofitamab, where we have the transient increases in IL-6.
Okay, thank you. There's another question by Armin. Do you mind if I just can you unmute, or do I need to ask?
Thank you. Well, you can just read it. It's all there. Yeah.
Okay. Is basically on glofitamab versus binatumomab, I don't know how to say that one. Yeah. The potency regarding triggering CRS. Could you comment on that? Any comments that basically, despite similar mode of action, the architectures of the molecules are different, including FC on glofitamab. Were there any differences regarding T cell activation and expansion during the treatment?
Yeah, maybe I'm thinking about the fact that in terms of the IL-6 levels, if we think about the IL-6 levels, the transient increase was similar between the two compounds. I mean, we are reaching this level of 1,000 picogram per ml following the first administration for both drugs. Maybe the difference that we will see will be, I was thinking about the PK, because the PK is much, the half-life is much shorter in the case of binatumomab. It's a matter of few hours, I think, as compared to days for glofitamab. But in terms of the increase of IL-6, I'm not sure there is specifically a difference. You are mentioning here, including FC on glofitamab. I'm not sure that this translates into huge differences in terms of IL-6 increase. I think that the experience is similar as well, if I remember well, for mosunetuzumab as well, where the levels of increase, at least they are associated when you look at the grades of CRS, grade 1, grade 2, for most of the patients. It's more or less a similar safety profile. Of course, it will depend on the step-up dosing or of the dosing regimen you select. But any comment on that, Neil? Not sure about.
No, I don't have anything additional.
Thank you very much. That's really useful to know, because part of the original message from Micromet obviously was that binatumomab is unique because it's so small that anything else just doesn't getting in the way in the immunological sign-ups will just abolish the effect. It's really interesting to see that the presence of FC actually doesn't make that much difference in that respect. Thank you.
Yeah, thanks. Yeah, I understand what you mean about the stoichiometry and the fact that you could attract more B cells to T cells when you have a smaller structure. At the same time, you have got this big difference as well in terms of half-life, as I mentioned, even though here it's not relevant, because we are talking, for example, about the first administration event. And
thank you.
Okay. I don't see any other questions coming through. So I'd just like to take the opportunity again just to thank Neil and Nassim for their excellent presentation, and congratulations as well on the publication. And I'm sure you've had lots of interest on the back of it. It's a very important topic, and congratulations as well on the labeling and the use of PBPK for that.
Excellent. Thanks, Leo.
Yeah, thank you. Do you mind just showing the last slide, and then we'll finish up. So the next webinar is on the 23rd of April. This one is obviously a different area now. We're looking at transporters or TP substrates, and this is by colleagues in Gilead who are going to be presenting on this. So please use the registration link. You can register on the web, on the members website. Okay, thank you everyone, and have a great day. Take care.
Thank you.
Thanks. Bye-bye.
Bye.
Bye.